Okay, we're running a few minutes late, but the last speaker of the session is Dr. Axel Petzold, who again wears several hats. He's at neurology and neurosurgery at UCLH and also at Moorfields Hospital and Amsterdam University Medical Centre. The most exciting thing is that last year, the Royal College of Physicians awarded him a prestigious Lady Estelle Wolfson Lectureship in Translational Medicine. So well done, Axel. That was good. Axel's talk follows nicely on from Palmer's because you're going to talk about neurofilaments and neurodegeneration. So that's great. Axel, please.
Thank you, and thank you, Sir Munir, for the invitation on your 10th annual meeting. It's nice to be here, and the two talks could not have been better combined because now you're all set up for neurofilaments, and I tried to structure the talk in a way to talk you into the subject, even if you don't know anything about neurofilaments. There will be an introduction on the protein, a bit on the experimental work, the clinical work, and I will focus on two trials before making some points of future discussions and directions which pick up on the questions which have been asked. So what I want you to show and to enjoy, hopefully, is a weekend I spent in Great Ormond Street, and I had a laser dissection microscope, and I had a neuron in a cell dish, and I was trying to cut through the membrane of the neuron just to demonstrate, people like you in the audience, why am I interested in measuring neurofilaments? Because what you can see is as the membrane of the neuron opens, that the cellular content is released, and that is where neurofilaments are then from the dying neuron released into the adjacent medium, be it the cerebrospinal fluid or the blood. And cutting an axon is very, very difficult. It's a tough structure for, I think, very, very good reasons. So if you try to make the next step in translational medicine, it was a proof of principle approach. So I used again a laser dissection microscope. And on the top of the slide, you see a sketch of a neuron with an axon, and the red bars represent neurofilaments. So it's stuck with neurofilaments, as Pam said. If you have, for example, a cell culture from your IPCs with a few neurons, you homogenize them, you get a bit of neurofilaments. And if you have a culture which worked much better with a lot of neurons, you homogenize them, you get a lot of neurofilaments, but that's very semi-quantitative. So what I've done in this experiment is just to use the same laser capture microscope, and you can see in the middle of the slide how I had a fixed neuron on a slide and cut it out. And if you collect different numbers of neurons, then you see a very nice correlation on the y-axis between the amount of neurofilaments you can measure and on the x-axis of how many neurons there are. And you need at least 46 neurons to get a signal, which is interesting maybe for in vitro experiments. And of course, the question is what happens in humans? Does it work? And we worked with traumatic brain injury. These were patients who did require, as their medical workup, a decomprehensive hemocranectomy. You see a bit of the bone missing on the CT scan, and they got a pressure monitor inserted. That's part of the management protocol of these patients. And adjacent to that, we had a microdialysis filter that's like a mini kidney, which then permits for the extracellular fluid adjacent to the neuron to be collected. And if you look at that over the time, that's the graph in the bottom, then you can see some varying levels of neurofilaments on 1 hour intervals. So it's very quick, which you can relate to clinical events, such as a rise in ICP. But the real question is, how can we take it from a proof of principle towards convincing people that it's a good marker for neurodegeneration? And at that time, the idea was just to take in an experiment a tissue block, where the top section of the tissue block goes into histology. Now, I did actually talk blindly without looking at the slides, did I? So in terms of the neurofilaments, there are 5 major forms. And it is relevant to the question on the different assays to realize that you have neurofilaments which are only expressed in the central nervous system. That's to the far right. That's yellow. That is internexin alpha. Then you have neurofilaments which are very convenient. You have a lot of them because they're expressed everywhere in the central nervous system and in the peripheral nervous system, to which light, medium, and heavy belong. And then you have peripherin on the far end in pink, which is only expressed in the peripheral nervous system. And I will come later back to why this may be important. What I have done in this study, it's, if you want to have a look up for all mutations, all post-translational modifications of neurofilaments, it's free for download with this QR code. But what I've done here is to show you with alpha-2 modeling how you use the neurofilaments like little pieces of Lego to build a structure. And that will become important. So the 5 monomers, they assemble to dimers. At that first level, nature's taken an important decision, and that is everyone must have light chain as a partner. That is why the light chain is so fantastic to be measured, because it's abundant. Different light is the only one which can self-assemble. You see that the 2 red parts down there, but every other one needs to pair up and tango with light, then to come into the tetramer. We have 4 of them. And once you have all of them, you arrange them in a circle-like structure, and then you put several of the circles above each other, which is called longitudinal annealing. And once you have all this string put together, you just close it and make it smaller by moving the side chains from the inside to the outside. And once you have this structure, you have a working neurofilament structure, which can be, by post-translational modification, changes in charge, interact with mitochondria and all that. So it's a long, long process of building it, but the light chain is a very useful, if you want, general marker. So there have been a number of generation of tests. All of you will be familiar with the immunoblots with these nice bands of the protein, but for quantification that is no good because it's semi-quantitative. So the first time ever we managed to quantify neurofilaments from various body fluids, the aqueous humor, the vitreous body, the blood, the extracellular fluid, the cerebrospinal fluid, the amniotic fluid, was using ELISA. The ELISA required still a large sample volume, and then came a new technology, electrochemiluminescence, and that is the MSD platform to which you referred, which reduced the amount of sample required by just improving sensitivity. But that was a short-lived period where MSD really was successful because it got superseded by the Simoa technology, which puts it down to the femtoliter range. And with that, the network of neurofilament research made really a breakthrough, because this is now measured on industrial scale level. And we are looking at the new platforms. There are 2, something which is used in the biobank on chip technology, gives you a lot of proteins, and then also point-of-care technique. We had earlier in the session this morning the question, how quick do we get a result? So the slide I spoke about before from memory is this one. So the setup of the experiment is to have a tissue block. The top section goes to histology, and you look at the structure of neurons and axons, and the bottom section gets homogenized, and you look at the biochemistry. And I will show 3 different experiments here. So to the very far left, that was the first experiment we did at UCL. We took the spinal cord from mice who had a degenerative condition called EIE. And if you compare the control mice to the far left with the EIE mice a little bit further right, then you can see in black, that's a silver stain for axons, for neurofilaments, that the axons in the affected animals are disrupted. So you have less of the axons. It's an important point I try to make about neurofilament levels, because if you now look at the homogenized tissue levels, those animals who had less neurofilaments had lower neurofilament levels than those who had intact axons and neurons. And that relationship between the amount of axons and neurons you have in the tissue and how much you measure can also be translated to humans. So you see in the middle of the slide, multiple sclerosis brain tissue post-mortem, and areas where you had plaques, they were void of axons. You have lower neurofilaments, the chronic lesions. And then, interestingly, in acute lesion, you even have an increase, and we can talk about that. But is this useful? And the neuron paper, I would have loved to have thought about this myself, is a wonderful study where the group in Tübingen introduced a mutation into mice which caused a neurodegenerative condition, that's in red. And if you then measure it in the blood, neurofilament levels go very high. Just to make this point, the two parts in the left of the slide look at the tissue, and the last study looks into the blood. So as neurofilament levels are reduced in the tissue because the neurons die, they increase in the blood. And what they showed in the neuron paper really beautifully is that treatment of these animals actually prevented or delayed this happening. And that is, in a nutshell, the concept of how brain tissue, nervous tissue, neurofilaments, and clinical events relate. In blue, I just give you the amount of brain tissue, spinal cord tissue, motor neuron tissue, if you want. At some stage, there is a clinical event, can be anything, trauma, stroke, you call it, and that kills neurons. So neurofilament levels increase in cerebrospinal fluid or blood, but then it takes some time until atrophy develops because you have an inflammatory response, you have some edema, and this time interval can be very, very long, but we can measure it accurately with structural imaging techniques. If you're later in the time course and there's another clinical event, the same repeats, but levels may differ a bit because your resources of neurofilaments, of course, of neural tissue are less. So, of course, neurofilaments have been measured in a large number of diseases, and just to talk you through the possibilities you have, and maybe that also involves some of the diseases people in the audience work on, much focus goes on dementia. In the moment we know we have approved trials, and of course neurofilaments and tau will be important. We heard from Pam a lot about motor neuron disease. Then in stroke medicine, neurofilaments are a very good marker of acute damage, and also that involves endarterectomy and other operations they do. There is a large body of literature on traumatic brain injury, and that is relevant because you alluded to also sport and athletes, and there has been a lot of research in ice hockey players, football players, and a little bit the message got out that these contact sports cause a raise of neurofilament levels, and I think the media largely interprets that as it equates immediate brain damage. So remember, the peripheral nervous system also has a lot of neurofilaments, and if you have just these concussions, the source of where the damage happened cannot be determined by neurofilament light because it's expressed in both compartments. So we need to be a bit careful about how to interpret that. A lot of data came in multiple sclerosis. These two diseases were both described by Charcot. And then we have a group of diseases which originally were multiple sclerosis and then through the discovery of antibodies turned into pneumonitis optica, MOG disease. Optic neuritis has a particular interest for me. Then we have a large range of neurodegenerative diseases, ataxias, Parkinson's disease. There is in pregnancy and pediatrics, so we heard about SMA. Neurofilaments are measured in SMA following these children up in pregnancy. Eclampsia, it will be used, for example, in some hospitals like Basel as a decision when to start birth. Then there is a big group here, infectious diseases. So we heard in the COVID pandemic that for septic encephalopathy, to which COVID belongs, neurofilament levels are elevated acutely in a bad prognostic symptom or sign, if you want. But now that we have the post-COVID situation, neurofilaments come back in there as a way of indirectly looking what type of brain damage these patients have. In oncology, now, I think that is for me one of the most interesting parts. because neurologically, I'm a neurologist, oncology means metastatic brain disease, and can neurofilaments tell you about that happening? But epigenetically, neurofilaments play a role, and I would have loved to know Professor Goodyear's opinion on it, play a role in the stabilization somehow of the genome. And in breast cancer, for example, it has an important epigenetic role in regulating oncology. And I think we need to learn more about that. Cardiologists have discovered neurofilaments as a way to guide them what to do about paroxysmal or atrial fibrillation, heart rhythm possibilities. And of course, in cardiac arrest, it is a prognostic sign if people have severe brain damage or not. So through networking, just as you have done here, through networking, the neurofilaments manage to involve all of medicine. But one of the reasons why I spoke so long about it is for you to understand it cannot and will never be a diagnostic test for one single condition, because it's expressed with all of them. So it is useful in a particular clinic context if you ask a good question, but you cannot have it as a diagnostic test. It's not specific. With that, I come to trials. I primed it. There are two diseases which Charcot in Paris did name. About one we already heard. The other one is multiple sclerosis, and this is another New England Journal publication on a new anti-CD20. And in fact, there are two trials put together. And what the graphs show is that you have a massive reduction of the lymphocyte count. But these are the graphs I want to put your eye to. This is another treatment drug, teriflunomide. We were not allowed to do placebo-controlled trial. It was not considered to be ethical. So what needed to be shown is that ocrelizumab as a new drug is superior to an existing drug. And if you see the neurofilament level on the y-axis and the time on the x-axis, these are the patients within that trial, a rapid decrease of neurofilament in both trial Asclepios 1 and 2. And that was the reason why the FDA gave rapid approval for this drug. It was based on neurofilament levels. And you already spoke about that, but Biogen put on their website that their drug got approved through the FDA based on neurofilament levels. So this is a massive step forward. You couldn't have put the scene up better. And you also alluded to this trial. In this trial, there is these are all individuals with a known mutation for motor neuron disease, and they enter two arms. One is like a conventional open label testosterone trial under 100 milligrams we just heard about, but this part is really interesting. So I highlighted in red. The neurofilament in the blood is taken every month, and if the levels rise above 44 picograms per milliliter, which is a little bit more than twice of the normal level, then a decision is taken to enter this really interesting phase of the trial, which is placebo-controlled. And the idea behind that is that if an earlier intervention based on neurofilament levels are going to be helpful or not, and if this works, it is a complete change, paradigm shift of how these trials are designed. Now, to put that in context again, I've shown it to you before. That's the tissue we have. Something happens, neurofilament levels in red increase, and the idea is, of course, if we do a clinical trial, that it reduces the damage so that we get lower neurofilaments, hopefully preserve tissue over a longer time, and that this is what does benefit our patients, because to go from high neurofilaments to low neurofilaments may be possible, but to get out of a wheelchair running again may be very, very challenging. So there are currently 281 trials which do use neurofilament levels registered, all range of diseases. And in the last 3 slides, this is in answer to your question on future developments. The first one is, I showed you my alpha modeling of different neurofilament forms. This is peripherin. So we just managed to get accepted into brain. I had hoped it would be online today, but maybe it's the end of the week. For the peripherin form, here you see the immunoblots, the first generation assays. That's the Neurofilaments light chain, and this is peripherin. And we just took it from this area of the spinal cord where the central and peripheral nervous system meet. If you then look at different diseases, Guillain-Barré syndrome is a peripheral nervous disease, acute, very elevated peripherin levels. And if you then look at some of the other diseases, on average lower, but there are some outliers, and there may be ways of us looking into peripheral nerve damage as well in order to find out what is going on there. Now the next slide is beautiful. The person who had done this, Gary Shaw, he originates in the UK and he went over to Gainesville. He's a phenomenal scientist. He was early in his career in Germany with Max Weber working on the expression of neurofilaments during development. So he was the person who told us that after birth we upregulate, for example, the heavy chain that induces myelination, and it all moves on. There's possibly no one in the world I know who knows more about neurofilaments than him. The one thing neither he nor I ever understood is why is it that this one antibody from Sweden, from the company called Human, Nicholas Norgren developed it, is so effective. Because we, of course, were interested in using these antibodies. And I remember sitting in the lab using this expensive antibody from Sweden, trying to stain neurofilaments, and I did not see anything. And now I thought maybe I did not store it, so I ordered again, and then maybe the temperature was not right, and it was just exasperating. But Gary had the same problem, except he has not given up. So what he has done is he has actually found out that if you do sort of the Fleming way of research and be favored by luck and leaving something a little bit. So what he has done is he stained the spinal cord of rodents, didn't see anything, and then he left them and went for a weekend away and came back and stained them again, and suddenly all the neurofilament light was in there. So he thought maybe something happens to these axons. So he did a series of percussion experiments and trauma experiments, and what he was able to demonstrate is really through patience and perseveration that what the antibodies do is they discover a proteolytic breakdown product which also has post-translational modification. So I'm not sure if it is citronated or phosphorylated, carbonylated, you name it. But what he has really done, he has pinned it down here. This is the sequence. This is where human body 2 and this where body antibody 1 stain, and in combination of that, the neurofilament light as it is released and modified is what the assay essentially captured. And I think that is incredibly important for the future of using these biomarkers, because the full-length protein is not very soluble. A proteolytic breakdown product is soluble. If you have a small proteolytic breakdown product, it's easier to equilibrate between different fluid compartments because it can diffuse through it. And if you then additionally find out high affinity antibodies to capture that, then you're in an absolutely perfect position to make use of this biomarker, which is so promising, not only to monitor what happens to our patients, but also to convince the FDA and other authorities that our treatments work. And with that, I come to the very last point. This is work from Professor Jens Kuhle in Basel, who is involved with a lot of trials. One of the points we don't like to talk so much about are the confounding factors, if you want. And one of the biggest one is age. So neurofilaments go up with age. You can see a lot of correlations with that. Another one is BMI. Another one is poor kidney function. And all that complicates the interpretation of neurofilament levels. So what Professor Kuhle has done was very, very intelligent. He constructed a Z-score, which you can download on your app. You can put everything in, you get the Z-score for neurofilaments, and that circumvents the problem of all these confounding factors. And I think that is where the statistical future lies in, because it makes it possible across different techniques, machines, laboratories, patient populations to compare it. And I heard he just got his other paper accepted also in the Lancet Neurology for the same Z-score in pediatric diseases. With that, I summarize it all. I started with a video just to share on a very, very simple level the biomarker hypothesis. Neurofilaments get released from the tissue, there's less in the tissue, you have more in the fluid you collect. And I tried to convince you that a good marker for neurodegeneration based on this translational approach. And the main point is about the clinical trials. They do not only serve as an endpoint, but with this new approach, they may even change a paradigm in how we design these trials. And then, of course, there are a couple of future applications. Look at post-translational modification, proteolytic breakdown, products diversify, look at different neurofilament isoforms. And with that, I do thank you for your attention.